LCAT and the CETP role in the HDL metabolism. The HDL, which is synthesized by the intestine, it is the nascent HDL. It is efflux into the bloodstream. It is discoid in shape because there is no cholesterol ester at this stage. The ApoA, ApoC, ApoE produced by the liver, they get binds to the HDL. In the peripheral tissue, there is the proteins called the ABC protein or cholesterol efflux regulator protein. What they do is it transfer the cholesterol from the cells to the HDL discs. It can transfer the cholesterol from the cells to the HDL discs. And the ApoA, one of the HDL, activates the LCAT, lecithin cholesterol acyl transferase. Then LCAT binds to the HDL discs. The HDL discs, in the HDL discs, in the bilipid layer, there is the presence of lecithin. At the carbon number 2, lecithin has polyunsaturated fatty acid. The LCAT, activated LCAT, what it do is it can transfer the polyunsaturated fatty acid from the carbon number 2 of the lecithin to 3-hydroxyl group of cholesterol, and that cholesterol becomes cholesterol ester, and the remaining lecithin remain as lysolecithin. Why it is needed to convert the cholesterol into the cholesterol ester? Why it is needed? The answer is the cholesterol ester is more hydrophobic and it moves to the interior of the HDL. It means it is packed more efficiently. That's why it is needed to convert the cholesterol into cholesterol ester. And second is the CETP. It acted in the transfer of cholesterol ester from the HDL to the VLDL LDL. That's why it needed to convert the cholesterol into cholesterol ester. Then how the LCAT named as? LCAT named as lecithin cholesterol acyl transferase. Why? Because it act on the carbon number 2 of the lecithin and convert the cholesterol into the cholesterol ester. That's why it named as lecithin cholesterol acyl transferase. And this process continue until the HDL completely filled with the cholesterol ester. That means the HDL becomes more spherical because the cholesterol ester moves into the interior part and the HDL becomes more spherical. That spherical HDL is called the mature HDL or HDL3. Then it gets internalized into the hepatocytes by two methods. Either it get directly internalized or it get transfer cholesterol ester to VLDL, LDL, IDL through CETP, and then it get internalized into the hepatocytes. There are two ways. The first one is it can directly internalize. Firstly, it can binds to the ApoA1 receptor. The ApoA1 present in the LDL, they can identify the ApoA1 receptor and binding and internalization occurs. But the point to note is that it is not a receptor that uptake the HDL. The main receptor that uptake the HDL is the hepatic scavenger receptor B1. It mainly helps in the internalization of HDL. When they get internalized, then hepatic lipase hydrolyze the HDL phospholipid anti-J, and the cholesterol ester release. The cholesterol ester release that get converted into the bile acids. This is and bile acid excrete as the bile. This is the only excretory excretion method of cholesterol. There is nothing else than this. And next one is the HDL through CETP pathway. How this pathway the HDL get internalized into liver? Firstly, the HDL3, the cholesterol ester present within it, the CETP present in the plasma. What it do is it can take off the cholesterol ester from the HDL3 and then transfer to IDL, VLDL as IDL in exchange of the TG. TG taken by the cholesterol ester transfer protein from VLDL, LDL, IDL and then transferred to HDL3. Now the HDL is rich in TG, that we call is HDL2. And then the HDL2 is internalized by the hepatocytes. Before it is get internalized, it is acted upon by the lipase, triglyceride lipase, hepatic triglyceride lipase. It is acted upon by the hepatic. It is hepatic triglyceride lipase, and then it is acted upon by the hepatic scavenger receptor. The above process is all of the process we call it the reverse cholesterol transport, because the cholesterol transport from the peripheral tissue to the liver through the HDL. And then the polyunsaturated fatty acid, we call it the anti-atherogenic. Why? Because it helps in the esterification of the cholesterol and helps in the removal of cholesterol. So it is anti-atherogenic because cholesterol is atherogenic. It helps in the atherosclerosis process, atheroma formation. It play a major role there. That's why it is anti-atherogenic. That much.